Hello everyone, welcome to another MCAT ACE lecture. So here we are doing the second part of the metabolic pathways that we've been describing for glycolysis, fermentation, citric acid cycle, and electron transport chain. These were all covered in the first lecture. Now in this follow-up lecture, which is the second part, we are now going to cover the pentose phosphate pathway, gluconeogenesis and glycogenesis, regulation of these pathway, and then finally conclude the lecture with metabolism of fatty acid and proteins and some practice questions in the end. So this is a high yield drawing that I've shown you at the beginning of the first lecture. So basically glucose is rich in potential energy, and we've discussed how through glycolysis and through aerobic respiration it can yield 36 to 38 ATPs. And then depending on the source that you're reading, whether it's NADH that is giving 2.5 ATP versus 3 ATPs, it could also be 30 to 32 ATPs. So the top one is assuming NADH gives 3 ATPs and every FADH gives 2 ATP. The lower one is assuming 2.5 ATP per NADH molecule and 1.5 per FADH molecule. That's going through aerobic respiration. Under anaerobic reduction, however, there would be only 2 ATP that is produced throughout the glycolysis. So we've already covered this topic extensively, describing how many ATPs, which stands for adenosine triphosphate, will be produced through each one of these reactions and the contributing factors that can affect the rate of all these reactions. Now, next we would like to discuss the role of glucose as a precursor for supplying different metabolic intermediates for biosynthetic pathway. So we will discuss how glucose can be used as a storage, can be in the storage form like glycogen, starch, or sucrose, or it can be used in the other pathways like pentose phosphate pathway for production of ribose 5-phosphate, which is required for DNA, RNA, and nucleotide synthesis. And this pathway also produces NADPH, which is required for synthesis of steroids, fatty acids, and the reduced form of glutathione, which is required for defense against oxidative stress. Now here I'd like to point out not to confuse NADPH with NADH. So if you recall glycolysis and then citric acid cycle, they were all giving us 10 molecules of NADH per 1 glucose molecule that was going throughout this reaction. Here we are talking regarding the pentose phosphate pathway, we are talking about NADPH. So this molecule has an extra phosphate and it's different than NADH. NADH is used for energy production or ATP / adenosine triphosphate production, while NADPH is used for different biosynthetic pathways. So that's one point I'd like you to pay attention to. And if you like to remember that pentose phosphate pathway is associated with NADPH, you can always think of pentose phosphate pathway is associated with production of NADPH. All right, now with that background information, let's move on to the actual pathway. So here we have the pentose phosphate pathway, the other name for which is hexose monophosphate pathway or shunt. So hexose mono pathway. So sometimes you hear the HMP pathway. That's what we are referring to, pentose phosphate pathway. And so here you can see that in this reaction, glucose throughout the glycolysis will be converted to glucose 6-phosphate. That's the first reaction, and that was via the hexokinase. These were all reviewed in the first lecture. Now, throughout the first reaction of the pentose phosphate pathway, the enzyme glucose 6-phosphate dehydrogenase will convert glucose 6-phosphate into different intermediates, as a consequence of which there will be production of 2 NADPH and 1 ribulose 5-phosphate molecule. And then the ribulose 5-phosphate will be later converted to ribose 5-phosphate, which is required for production of nucleotides, RNA, and DNA. while the NADPH is used for production of the reduced form of the glutathione, steroids, as well as fatty acids. Now, one more point about the glucose 6-phosphate dehydrogenase. Not only this is the first reaction of the pentose phosphate pathway, this is also the rate limiting step. And so if NADP plus concentration is too high, then it would have a positive effect on the glucose 6-phosphate dehydrogenase in order to produce more NADPH. And then if there is too much NADPH, then there would be a negative effect in order to lower the production of the NADPH. Now, if you've noticed, the reduced form of glutathione is being produced via the G6PD reaction. So glucose 6-phosphate dehydrogenase is required for production of GSH, which is the reduced form of glutathione that is helpful in defense against reactive oxygen species. So what happens in patients who don't have this enzyme? Therefore, they would be susceptible to attack by reactive oxygen species, and so these patients can develop hemolytic anemia from any compounds that can cause reactive oxygen species, like for instance sulfa drugs can cause hemolytic anemia in these patients. Alright, now moving on with the pentose phosphate pathway, there is actually two different phases. One is the oxidative phase, which I just described for you, and the net reaction is glucose 6-phosphate plus 2NAD plus H2O will yield ribose 5-phosphate, a CO2 molecule, and 2 NADPH plus 2H plus. So this is basically the net reaction. I'm just going to draw the reaction in a little more detail for you. But then there is also a non-oxidative reaction where the ribulose 5-phosphate can be converted back to the glucose 6-phosphate. So if you have too much production, there is a way of regenerating the initial molecule, and this is called the non-oxidative phase, and I'll go over that in a second. But first, let's go over the oxidative phase. Alright, so here we have the starting molecule, glucose 6-phosphate. There are a few reactions that are going on. The first one is carried out by glucose 6-phosphate dehydrogenase enzyme, and then these first reactions will give rise to 6-phosphogluconate. And then later on, 6-phosphogluconate will be converted into ribulose 5-phosphate, and now this can be converted into ribose 5-phosphate, which is essential for RNA, DNA, and nucleotide production. And then in the first reaction, there will be water that will be going in, and an NADP plus molecule that is converted into NADPH. And then likewise, in the reaction shown here, another NADP plus will be converted to NADPH, and a CO2 will be released. So here you can see that 2NAD plus is converted to 2NADPH, an H2O molecule has entered the reaction, and then a CO2 is released. Alright, and then the net outcome is a ribose 5-phosphate. So now, how does NADPH help in the conversion of oxidized form of glutathione? So the oxidized form of glutathione is shown as GSSG, and so NADPH can be converted back to NADP+, and throughout this reaction converting the oxidized form of the glutathione into the reduced form, GSH. So it's like a circle. NADP+ converts to NADH, and then by its conversion back to NADP+, an unoxidized form of glutathione will become a reduced form. And then all these reactions also have an H plus H plus H plus, so that's how we get that 2H plus there. Alright, so that's with the oxidative phase. Now, we also have a non-oxidative reaction which converts the pentose phosphate back into the hexose phosphate. So the ribulose 5-phosphate will be converted back into glucose 6-phosphate. And so this mechanism provides a mechanism in order to regenerate the initial molecule if too much products have been produced here. So we had the glucose 6-phosphate through a series of reactions has been converted into ribulose 5-phosphate. Now via two different enzymes called transketolase and transaldolase, this pentose phosphate is converted back into the hexose phosphate, and that's the non-oxidative phase of the pentose phosphate pathway. Now, we keep talking about the reduced form of the glutathione, GSH, and how it can help protect against oxidative stress or oxidative damage. The outline of the MCAT mentions oxidative damage as one of the topics that you're required to know for your exam. So now is a good opportunity to go over the oxidative damage since we are on the topic. So let me first describe for you how oxidative damage can be initiated. So basically conditions like, for instance, mitochondrial respiration, ionizing radiation, certain drugs like, for instance, sulfa drugs or anti-malarial drugs, these can convert the O2 into superoxide radical, O2 negative. And then later on, the superoxide radical, so this is called superoxide radical, can be converted into hydrogen peroxide. Hydrogen peroxide. This of course required 2H plus and an electron to enter the pathway. Now H2O2 can then, via H plus going in and losing an H2O and an electron entering, can be converted into hydroxyl radical, which is extremely toxic. And by being toxic, the reason for that is that this is highly unstable, and so therefore it would right away react with lipids, DNA, and proteins. I shouldn't really say it's toxic, it's not really toxin, but it's a molecule, unstable molecule that will damage all these structures, lipids, DNA, and proteins. And so therefore it would cause mutation, for instance, in DNA, as a consequence of which now there will be problem in the cell and can lead to apoptosis. Now, how can we fight oxidative damage that is induced by all these mechanisms? Because mitochondrial respiration is part of the requirement for aerobic respiration and energy production in mammals and all other aerobic organisms. So we have different enzymes that helps break down these molecules. For instance, the superoxide dismutase enzyme helps convert the superoxide radical into H2O2. And then hydrogen peroxide can be broken down by two different enzymes, catalase, and the other enzyme is the glutathione peroxidase. And I'd like to focus on glutathione peroxidase here. So here we have two reduced form of the glutathione. They will react with H2O2, and throughout this reaction, two molecules of H2O will be produced. So that's the mechanism through which the glutathione peroxidase can neutralize the hydrogen peroxide. Alright, so that's about it of the topic of pentose phosphate pathway and how it would help produce the reducing form of the glutathione that helps defend against reactive oxygen species. The next topic is gluconeogenesis. So this is a high yield diagram here that shows you how different pathways can lead to production of glucose. So glycogen breakdown, we already discussed, it will lead to production of glucose. Triglycerides will produce glycerol and fatty acids, both of which will contribute to either the products of the glycolysis or acetyl coenzyme A. And acetyl coenzyme A, it cannot right away go back through the same mechanism as glycolysis. 7 of these reactions are reversible, but then the others are not. And so through some other bypassing reactions, acetyl coenzyme A is able to get converted back, up to the glucose, converted back to glucose. And I'm going to review those reactions with you. But my point is that acetyl coenzyme A, in order to get converted back to glucose, some of the reactions of glycolysis are reversible, some are not. But it's possible to convert acetyl coenzyme A back into the glucose. We also have proteins that will break down into amino acids, and then they can contribute to either pyruvic acid, acetyl coenzyme A, or citric acid cycle, and then again acetyl coenzyme A can be converted back to glucose. And then finally we have the lactic acid here that can be converted into pyruvic acid, and now it can get converted to glucose again. Alright, now what's the point of producing glucose? Why is it so essential to produce glucose? And so basically glucose is the main fuel and building block for many, I can't say for all organisms, but for most organisms, ranging from microbes to humans. But then the supply of glucose and glycogen may run short under certain conditions, like between meals, during longer fasting periods, or after vigorous exercise. And so that's the time that lipids or proteins or lactic acid or other pathways will lead to production of the glucose. And this process is called gluconeogenesis, means production of glucose. Now regarding the lactic acid, I'd like to mention that glucose in the muscle will be converted into pyruvate, which will be converted into lactate inside the muscle. Now this lactate, through the bloodstream, will get transferred to the liver. So here we got the liver, here we had the muscle, and here is the blood stream. So now we have the lactate transferred to the liver, it will get converted back to the pyruvate, and then glucose through the process of gluconeogenesis, and now glucose can again enter the muscle and go through glycolysis and produce energy, and this cycle continues to and the cycle continues. And so this cycle is referred to as Cori or lactic acid cycle. Alright, now next move on to the regulation of glycolysis and gluconeogenesis. So basically these two pathways, they must be reciprocally regulated so that once one pathway is active, the other one remains inactive. Otherwise, if both are active, one is producing glucose, the other is destroying glucose. If we keep going back and forth, the only outcome is heat production. So in order to be effective in a different biological system, once one of these mechanisms is active, the other one should be inactive. And this is due to the fact that the two pathways are running in different direction. So here you can see that glucose, in order to get converted to pyruvate, that would be glycolysis. But then conversion of pyruvate back to the glucose, that would be now the gluconeogenesis. So they're exactly in reverse directions. And so once one is active, the other one should be inactive. Now, in order for gluconeogenesis to occur for pyruvate back to the glucose, some of these glycolytic reactions are reversible, but then there are certain ones that are not reversible, and so those ones I've just listed them here as red enzymes. So here you recall for glycolysis, phosphoenol pyruvate could be directly converted to pyruvate. In order for the reverse reaction to occur, pyruvate has to be converted into oxaloacetate, which is one of the intermediate products of the citric acid cycle, and then this reaction is carried out by the pyruvate carboxylase enzyme. And next, oxaloacetate is converted to phosphoenol pyruvate by the PEP carboxylase enzyme. So that's the first two enzymes. And then finally, for conversion of fructose 1 and 6 biphosphate into fructose 6-phosphate, we need the fructose 1 and 6 bisphosphatase enzyme. Now tell me, what was the rate limiting enzyme of the glycolysis reaction? It was the phosphofructokinase 1, which was converting fructose 6-phosphate into fructose 1 and 6-phosphate. So this reaction here, phosphofructokinase 1, was the rate limiting enzyme, and this is not reversible, and so we have to use the fructose 1 and 6 bisphosphatase. And then the final reaction is glucose 6-phosphatase to convert the glucose 6-phosphate back to the glucose. Now the important point I'd like you to know here is that for gluconeogenesis, fructose 1,6-bisphosphatase is the rate limiting step, while for glycolysis, which was in this direction, phosphofructokinase 1 is the rate limiting step. So therefore that's why once one of these enzymes is active, the other one is in a relative inactive phase. And I'm going to explain for you how that works. So here is a nice drawn pathway for gluconeogenesis and glycolysis. For gluconeogenesis we have to go in this direction, while for glycolysis we go in the other direction. And these enzymes are being controlled by insulin and glucagon. Now remind me again, what is the function of insulin? The function of insulin is to decrease the blood glucose level. So if its function is to decrease blood glucose, therefore we don't need gluconeogenesis. So therefore insulin will push the pathway towards glycolysis. Glucagon, on the other hand, its function is to increase the blood glucose concentration. And so therefore, once there is a glucagon secretion, there would be pushing of the reaction towards the gluconeogenesis. So the fructose 2 and 6 bisphosphatase will be activated by this hormone. There is also this intermediate molecule called fructose 2 and 6 bisphosphate, which can regulate the activity of these two enzymes. So during the fed states, there is increased production of fructose 2 and 6 bisphosphate, as a consequence of which it would promote activity of phosphofructokinase 1, and so glycolysis will increase. And that makes sense, because during the fed state we already have too much sugar in our blood. We don't need more gluconeogenesis, so the pathway would be pushed to the right side. On the other hand, during starvation or fasting state, there would be drop in the concentration of fructose 2,6-bisphosphate, and so now reaction will be pushed to the left side in order to increase gluconeogenesis and increase the blood concentration of glucose. And here is a nicely drawn pathway for you. It shows you that in the fed state there is increased concentration of the enzyme phosphofructokinase 2 that converts fructose 6-phosphate into fructose 2,6-bisphosphate. This is the same molecule that we just discussed right here. Alright, and this molecule enhances activity of phosphofructokinase 1 in order to promote glycolysis. On the other hand, in the fasted state or starvation state, there is increased activity of fructose 2 and 6 bisphosphatase, which breaks down fructose 2,6-biphosphate into fructose 6-phosphate. And so since the concentration of this molecule has now decreased, therefore the reaction will be pushed to the left side in order to increase gluconeogenesis and glucose production. And here I have a nice memory aid for you to remember that anytime you eat at PFK, your blood concentration of PFK increases. So if there is increased PFK or increased PFK, as you can see here, therefore there would be increased rate of glycolysis. And it makes sense. Again, as you eat, what happens to your blood sugar? Your blood sugar increases. You don't need more gluconeogenesis. You need to break down glucose now. So therefore, once you're fed, there is increased concentration of PFK and there would be increased glycolysis. All right, so that covers the gluconeogenesis pathway. Now next, I'd like to discuss the glycogen synthesis. So basically, glycogen is primarily stored in liver and skeletal muscles, and its job is to maintain blood sugar and meet the energy demands of the muscles. So here in the liver, it helps maintain the blood glucose level, and in the skeletal muscles, the energy demand. And so the glucose synthesis can happen in 3 steps that are listed right here. So step 1 is carried out by the enzyme uridine diphosphate glucose phosphorylase. So uridine diphosphate glucose phosphorylase, which catalyzes formation of UDP glucose. So you can see here that glucose 1-phosphate is combined with uridine triphosphate to yield uridine diphosphate glucose plus the phosphate groups there. So now that we have the uridine diphosphate glucose, the glycogen synthase enzyme can catalyze the formation of polyglucose. So here we have a glycogen molecule that has N residues of glucose. So here we have N number. And via the glycogen synthase, now this new sugar molecule can be added. And so the number increases to N plus 1. So now we have another one added. We have N plus 1. And then we also have the UDP as another product. And then finally, in order to make branches, we need a third enzyme, and that's the step 3 that is listed here. So here we have the enzyme amylo 1 and 4 to 1 and 6 transglycosylase, which catalyzes the branch formation by cleaving the alpha amylose chain at the 1 and 4 linkage. So here we can see that we have the number 1, 2, 3, 4, 5, 6, and then carbon 1, 2, 3, 1, 2, 3, 4, 5, and 6. So here the enzymes breaks the bond between alpha 1 and 4 linkage and forms a bond between alpha 1 and 6. And so this way there will be a branch formation in the glycogen molecules. Next we have the glycogen degradation. So when we have too much glucose, we form glycogen. When we don't have enough, then we will have to break it down, either in the liver to raise the blood sugar or in the muscle in order to provide enough energy. And the glycogen degradation is being carried out by 3 different enzymes that are listed here. So I've briefly touched on this topic in our earlier lecture, but now here I'd like to go in more details describing every one of these reactions. So the first enzyme is glycogen phosphorylase, which catalyzes phosphorylasis of the glycogen molecule and breaking all these alpha 1 and 4 linkages. So as it does that, the glucose 1-phosphate will be released from the glycogen molecule. Now, glucose 1-phosphate is not really an intermediate of the glycolysis. We will need glucose 6-phosphate. Remember, we had glucose and in the first reaction of the glycolysis, it was being converted to glucose 6-phosphate by the hexokinase enzymes. So this is what we will need, and so therefore now we have the enzyme called phosphoglucomutase, which will convert the glucose 1,6-phosphate into glucose 6-phosphate. Alright, so that's breakage of all these alpha 1,4 linkages. Now how about the alpha 1,6 linkages? In order to do that, we will need the function of the debranching enzyme, so glycogen debranching enzyme. So the glycogen debranching enzyme functions via the activity of two different enzymes. The first one is the glycosyl transferase, which will move all these glucose residues to a nearby chain. So it will translocate them. Here you can see that the green ones have been moved to the nearby chain. So what we are left with now is only the alpha 1 and 6 linkage. And so this bond can now be broken by the alpha 1,6 glycosidase enzyme, which will release this hexose molecule. Alright, so that's it. That was the discussion of the glycogenolysis pathway. Now we'd like to discuss how this pathway is being regulated. So there's two modes of regulation. One is by allosteric effectors, and the other ones is by hormones. And so these allosteric effectors, they would mediate changes in the rate of glycogenolysis at the cell level, while hormones affect the whole organisms at the organismal level. So now, what are some of the allosteric molecules that would reduce the rate of glycogenolysis? So as we discussed in our first video, I don't want you to memorize these molecules. I want you to know the concept. So here I'm going to give you some example, like for instance ATP, glucose 6-phosphate, and glucose. All these molecules inhibit the function of glycogen phosphorylase. And why is that? Because we already have too much ATP from glycolysis, and we have too much glucose and glucose 6-phosphate here. So we don't need glycogen to break down more, because already all these pathways are saturated. So let's hold off on glycogenolysis and save this molecule for the time that we need it. So here, let me show it in the pathway here. So there, glucose, we already have too much ATP, so it's telling us that our energy is doing good. And then we also have too much glucose here. So why do we have to break down glycogen? We don't need to break it down. We already have too much glucose. We have already too much glucose 6-phosphate. We already have too much ATP. And so therefore, all these allosteric effectors will inhibit the function of glycogen phosphorylase in order to inhibit breakdown of glycogen. Alright, on the other hand, some molecules, like for instance AMP, tells us that we don't have enough energy, and so therefore it would change the conformation of the enzyme into its active form. So AMP will activate glycogen phosphorylase in order to break down glycogen into more glucose molecule, and now glucose can undergo glycolysis and produce more adenosine triphosphate. Alright, so that was the effect of the allosteric effectors. All these effects are at the cellular levels. Next, we would like to discuss different hormones that are affecting at the organism level. So now, regulation of glycogenolysis by different hormones. The first one that I'd like to discuss is insulin. So remind me again, what's the function of insulin? The function of insulin is to decrease blood concentration of glucose. So we don't need any more glucose. So what insulin does is that it inhibits the glycogenolysis, because we don't want any more glucose. We don't want glycogen to break down. and then glucagon, epinephrine and norepinephrine, and then glucocorticoids. So here insulin decreases glycogenolysis, and so therefore it decreases the blood glucose, while all these other increase glycogenolysis, as a consequence of which there would be increased blood glucose. Now every one of these hormones act through different mechanisms, and sometimes examiners like to provide you in the passage different mechanisms and then ask you about the details of those. So here I thought it would be a good idea just to quickly review some of these mechanisms for you so that you would be familiar with what might show up on some of the passages. Alright, so first starting with the insulin, it binds to its cell receptor and activates the protein kinase, which will cause phosphorylation of the signaling molecules. And these signaling molecules will activate glycogen synthase and inactivate glycogen phosphorylase so that glycogen does not break down anymore. Then we have glucagon, which increases the activity of the adenylate cyclase, as a consequence of which now there would be increased concentration of cyclic AMP that increases the glycogen breakdown, and so therefore it increases the glucose concentration. Epinephrine and norepinephrine, they function by two different mechanisms. The first one is that they increase cyclic AMP concentration, and so therefore they increase glucose concentration. And then the second mechanism is that they stimulate the phospholipase C enzyme, which will produce second messenger molecules such as inositol 1,4,5-triphosphate. So inositol 1,4,5-triphosphate, and it's shown as IP3. Diacylglycerol and calcium molecules. And what these second messengers do is that they reinforce the cell's response, so heighten the cell's response to cyclic AMP, as a consequence of which now enhance glycogen breakdown and increase glucose concentration. And then finally we have the glucocorticoids, and the effect of glucocorticoids is tissue specific. So here, so it depends which tissue we are talking about. If we are talking about the liver, inside the liver, glucocorticoids enhance both gluconeogenesis and glycogenesis. So increases concentration of both glucose and glycogen. But then if you're talking about skeletal muscles and adipose tissues, so muscles and fat, the glucocorticoids decrease insulin sensitivity, and so therefore they decrease glucose uptake and glycogenesis is also reduced. And so that's why by excess glucocorticoids exposure there would be insulin resistance and hyperglycemia that is seen in type 2 diabetic patients. So here is summarizing these pathway. That's basically generalizing how they work, if there is high blood glucose, then insulin need to kick in order to enhance glycolysis, glycogenesis, and lipogenesis. So any pathway that would destroy glucose or turn it into something else would kick in order to reduce the blood concentration of glucose. So here, if we go to the original pathway, we have too much glucose. So therefore, glycolysis will be increased, storage will be increased, pentose phosphate pathway, all these other pathways will kick in order to reduce the blood glucose. And these are all activated by the insulin. On the other hand, once we have low concentration of blood glucose, then at that time the hormones cortisol, glucagon, or epinephrine kick in to increase the glucose production, increase the glycogen breakdown, and increase the lipid breakdown. So far we've covered gluconeogenesis and glycogenolysis and glycogenesis. Next we would like to move on to the lipid metabolism and then finally into the amino acids. So here was our initial outline. We've already discussed the pentose phosphate pathway. We've discussed the gluconeogenesis, glycogenesis, glycogenolysis, and the regulation of these pathway with either allosteric effectors or hormones are discussed. So now we are on the last topic, which is the metabolism of fatty acids and finally proteins and how they would contribute to energy production. Alright, so let's move back to where we started. Alright, lipid metabolism. So basically lipids are hydrophobic molecules that are stored in the fat, or the other name for which is adipose tissues, and its storage form is triacylglyceride. So triacylglyceride, also known as triglycerides. These molecules consist of 3 long-chain fatty acids and a glycerol molecule. Now, fatty acids are an important source of energy production. So here in this high-yield pathway, I've shown you how fatty acids can contribute to the production of acetyl coenzyme A. And so where do we get our fatty acids? And so the source of the fatty acids could be either from diet, which I've listed here, number 1, or it could be obtained, it could be synthesized from sugars, amino acids, or other fatty acids, and I've listed here number 2. So let's go through those now. So digestion of dietary fatty acids starts in the small intestine. Bile salts help break the fat globules into smaller particles called micelles. And so this is done with the help of bile salts, as we just discussed. So here I have a drawing for you in order to make the understanding of this concept easier. So we have the fat globules that are all clumped together. In order for the digestive enzymes to work, for the lipase to break them apart, we need to increase the surface area, because as is, the lipase doesn't have really much space to penetrate. And so that's where the bile salts come into effect. And bile salts are amphipathic molecules, meaning that they have both the hydrophilic region and hydrophobic region. And so the hydrophobic areas will surround the micelles, while the hydrophilic areas will be exposed to all the water that is outside in the intestine. And so this way you can see that the fat globule has been broken down into many different micelles, and this process refers to as fat emulsification. So here we have the emulsified micelles. Now, one question that I have for you here is that what's the difference between bile salts and bile acids? So we know that cholesterol is being converted into bile acids in the liver. But then what are bile salts? So basically, once bile acids conjugate to glycine or taurine, the conjugated form of bile acids are called bile salts. And the purpose of this conjugation is that conjugation of glycine or taurine decreases the pKa of bile salts, as a consequence of which, now because the pH of the intestine is higher than the pKa of these molecules, they would be ionized. and so they would function more effectively in separating the micelles from the fat globules. Alright, so moving on. Digestion of dietary fat in the small intestine with the bile salts breaking the fat globules into smaller particles called micelles, and the purpose of that is to increase the surface area. At this time, the lipase hydrolyzes the triglycerides into its free fatty acids and glycerol. From there on, there would be absorption of these molecules through the epithelial cells of the intestine. Then they would convert back to the triglycerides, bind to the lipoprotein to form the chylomicrons. And the function of now these chylomicrons is to transport the fat to the lymphatic system, and then later on in the bloodstream, and then from the bloodstream now the fat can be delivered to different tissues. And we are going to cover different lipoproteins and their function in distribution to different body parts like LDL, VLDL, HDL, and so on. But here what I would like you to know is that chylomicron was the lipoproteins in the small intestine that have helped transport the triglyceride through the lymphatic system and the bloodstream. Alright, so that's one way of obtaining the fatty acids from the diet. The second way is creating our own fatty acids. And so fatty acids can be synthesized in the cytosol from acetyl coenzyme A, which is a product of the amino acid degradation, fatty acid oxidation, or glycolysis. So here let me show you what I mean. So acetyl coenzyme A can be converted back to fatty acids. And so what it was saying that acetyl coenzyme A can be product of the fatty acids, pyruvic acids, from the glycolysis, or the amino acids. Alright, so now the first reaction is the acetyl coenzyme A carboxylase mediating the conversion of the acetyl coenzyme A molecule into malonyl coenzyme A, which is a 4-carbon molecule. Alright, so here you can see the reaction. Acetyl coenzyme A, by addition of an HCO3, is now converted into malonyl coenzyme A. And then the next enzyme is the fatty acid synthase, which combines 7 malonyl coenzyme A with an acetyl coenzyme A, releases 7 CO2 in this process to form the palmitate, which is a 16-carbon molecule. So let's make sure I got it right. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, and 16 molecules. Now throughout this whole process, we can summarize the net reaction as 8 acetyl coenzyme A. So here we had 7 acetyl coenzyme A converting to 7 malonyl, and then here is the 8th one. So the net reaction is 8 acetyl coenzyme A combining with 7 ATP, 14 NADPH, gives rise to the palmitic acid, alright, which is a 16-carbon chain. So here, when I'm showing you that the acetyl coenzyme A can be converted to fatty acids, that's the mechanisms I'm describing. And when I'm showing you that fatty acids can be converted to acetyl coenzyme A, that's the breakdown process, which I will discuss next. Alright, now the last point that I'd like to mention before moving on to the fatty acid degradation is the rate limiting reaction. So basically acetyl coenzyme A is the rate limiting enzyme of the fatty acid synthesis, and it's therefore upregulated by citrate and insulin. So what do these reactions tell us? If there is too much citrate, we already have too much molecules for undergoing citric acid cycle. Or if you have too much insulin, it means there's already too much glucose in our blood, so we don't need glucose. We can convert them into fat. So that's why if there is increased citrate or increased insulin, there would be enhanced activity of the acetyl coenzyme A carboxylase to increase fatty acid production. On the other hand, if we have the other molecules, like for instance glucagon and epinephrine, which their function is to increase the blood glucose, they would inhibit the activity of acetyl coenzyme A because they don't want to store the valuable resources into fatty acids. They'd rather push the mechanism through gluconeogenesis in order to produce more glucose. So these hormones will inhibit fatty acid production. Alright, so that was with the fatty acid synthesis. Now let's go through fatty acid degradation. So basically, the reason fatty acids are so valuable is that they can act as a source of energy, and they function by removal of 2 carbon fragments in the form of acetyl coenzyme A. And so this reaction is carried out in 2 steps. In step 1, the acyl coenzyme A transferase enzyme attaches the coenzyme A to the fatty acid molecule. So here, let's say that we have a fatty acid O negative. Now with this acyl coenzyme A, we will have S coenzyme A replacing it. And now in the second reaction is where beta oxidation kicks in. So here is the product of the first reaction, acyl coenzyme A, and now acyl coenzyme A will break down into a shorter molecule of acyl coenzyme A, which is 2 carbon shorter, and an acetyl coenzyme A. And now this acetyl coenzyme A can be functioned in many different processes. It can undergo gluconeogenesis, or it can undergo citric acid cycle and electron transport chain in order to produce more adenosine triphosphate. Alright, now here, if you've noticed, I've written all the reactions for you. I'm not going to go through the mechanism of organic chemistry or chemistry. Already our chemistry professors have done a great job explaining these pathways and mechanisms. So here I'd like you to focus on the biology aspect of beta oxidation. So 2 different enzymes were involved in fatty acid degradation: acyl coenzyme A transferase and beta oxidation. And then each round of beta oxidation throughout the reactions that I've listed right here, you can see that they would produce 1 molecule of NADH, 1 molecule of FADH2, and then we already discussed 1 acyl coenzyme A. So now we have so many valuable resources for production of energy or glucose by beta oxidation. Now, a few other points I'd like to mention here is, so far we've been assuming that the molecule that is undergoing oxidation has an even number of carbons. But what if we have an odd number fatty acid? What happens then? So basically the same reactions goes through, except that the last cycle produces 1 molecule of acetyl coenzyme A and 1 molecule of propionyl coenzyme A, which is converted into succinyl coenzyme A, which is a TCA intermediate molecule, or alternatively it can be used for the gluconeogenesis production. Now, the other condition that we may have is unsaturated fatty acid. The mechanism that I've shown you here was for saturated fatty acid, but what if we have unsaturated fatty acid? What happens then? So the cis double bond is converted to the trans by the NOL coenzyme A isomerase, and this reaction basically replaces the first step of beta oxidation. So our first step here is now replaced with this new one in order to convert the cis into the trans bond. And so therefore there would be one less FADH2 molecule. So for every unsaturated bond that we have here, there would be one less FADH2 molecule that will be produced from the beta oxidation of fat. Alright, next we have ketone bodies. So acetyl coenzyme A can also be used for production of ketones. And here again, this extremely high yield image diagram that I have for you. Let me get rid of all the written stuff here. So there you go. You can see that acetyl coenzyme A can also be converted into ketone bodies, and that's what we're discussing next. So ketone bodies are primarily found in the mitochondria of the liver, and they function as a fuel for heart, skeletal muscles, and the brain. And these are and ketone bodies are produced during the condition of starvation and glucose shortage. So if there is starvation or high fat diet, as a consequence of which there is not enough carbohydrates, therefore ketone bodies will be formed. Ketone bodies are also seen in type 1 diabetic patients. And so these patients have hyperglycemia, too much glucose in their blood. Could you explain to me why would ketone bodies be formed? So remind me again, what's the difference between type 1 and type 2 diabetic? Type 2 are insensitive to insulin, while type 1 is not producing insulin. So since there is no insulin in type 1 diabetes, therefore even though there is so much glucose in the blood, it cannot get absorbed into the muscles. So the body perceives its condition as a condition of starvation because all these muscles don't get any glucose, and so it would increase the production of ketone bodies. And if you see ketone bodies in type 1 diabetic patients, it's called diabetic ketoacidosis. And so here it's showing you how acetyl coenzyme A can be converted into acetoacetate, acetone, or beta hydroxybutyrate, all of which are considered as ketone bodies. Now patients with diabetic ketoacidosis are said to have fruity breath, and this is because their breath has smell of acetone, and so that's why they say they have a fruity breath. Alright, now moving on to the last topic, which is protein digestion. So basically digestion of proteins start in the stomach, where the hydrochloric acid will activate the pepsin, and pepsin will hydrolyze the peptide bonds. Now digestion continues in the small intestine by the trypsin and chymotrypsin, which hydrolyze peptides into amino acids. And then later on, amino acids will enter the blood for transport to the liver, where at this site amino acids are degraded into ammonium, which is converted to urea for excretion. So here you can see that the proteins are converted into amino acids. The carbon skeleton can be used for production for the citric acid cycle. It can be used for production of acetyl coenzyme A or pyruvate for glucose production. So this is the gluconeogenesis. And then acetyl coenzyme A, we already discussed, it can be used for fatty acid production, it can be used for ketone bodies, it can be used for citric acid cycle. And so that's all for the carbon skeleton. And if you need a reminder, let me show you again this high yield drawing here. So proteins are broken down into amino acids. The carbon's backbone can be converted into acetyl coenzyme A, pyruvic acid, or go into the citric acid cycle, while the ammonia is converted into urea and excreted in urine. Alright, so here ammonia converted to urea and excreted in urine. And the type of, and here I have the reaction written down for you. So breakdown of the amino acids will yield CO2 and NH4 plus 2 ATP via the enzyme carbamyl phosphate synthase 1, which is the rate limiting step. The carbamyl will be formed, carbamyl phosphate, it would enter then the uric acid cycle, which gives out urea that can now get excreted in the urine. Alright, so we are basically done with this lecture. Now I'd like to cover some of the other topics that I didn't want to disrupt the flow of the lecture, so I saved them for the last. Let's discuss those, and then we will conclude the lecture with some practice questions. So if you give me a second, I'm going to pull up those questions. Alright, there we go. Now, the first topic is, what is lactose intolerance? So basically, certain individuals don't have lactase activity in the intestinal cells, as a consequence of which lactose cannot be processed until they reach the large intestine, as a consequence of which they would increase the osmolarity of the intestinal content and lead to water retention and diarrhea. And so that's why patients that are lactose intolerant, if they drink milk, they develop diarrhea because of this mechanisms that we just described right here. Now, how do thyroid hormones affect the rate of glycolysis and gluconeogenesis? So thyroid hormones are basically increasing the basal metabolic rate, as a consequence of which they would increase rate of glycolysis, they would increase the rate of gluconeogenesis, they would increase absorption of glucose in the intestine, increase protein synthesis, increase heart rate, cardiac output, and so therefore increase blood flow to different tissues. How do obese patients develop insulin resistance? And then what's the difference between type 1 and type 2 diabetes? So in patients that are obese, there is increased production of the insulin in order to compensate for elevated blood sugar, as a consequence of which, since there is now increased number of insulin products there, therefore the number of insulin receptors decrease, and so now there would be insulin resistance. And so therefore exercise and weight loss can help adjust the blood sugar, so it would decrease the blood sugar and restore insulin sensitivity. And so that's why they say type 2 diabetes. If you have an obese patient, if they exercise and lose weight, they could get cured and their diabetes go away. That's the mechanism we are talking about, to restore the sensitivity in insulin. Now the next question is, what's the difference between type 1 and type 2 diabetes? We've already discussed. With type 1, there is autoimmune destruction of the beta cells that are producing insulin. So there is no insulin production. While with type 2, it's usually seen with obesity due to increased resistance to insulin. Now, there is increased resistance, but there is still some insulin production in these patients. So therefore, patients with type 2 diabetes don't develop diabetic ketoacidosis. While in type 1, since there is no insulin whatsoever, the body perceives its condition as starvation state, because none of the sugar in the blood can get into the muscles, and so therefore increases the ketone body formation and cause diabetic ketoacidosis, which presents with a fruity breath odor. And the other term that you may see is Kussmaul respirations, particularly in medical schools. This is very important and high yield, and these patients have rapid and deep breathing pattern in order to compensate for the acidosis that increased production of ketone bodies has caused in their body. And then of course, nausea, vomiting, and altered mental status is some of the other signs of diabetic ketoacidosis. Next question, what is the function of brown fat? So basically, brown fat, it has a special protein in its mitochondria inner membrane that allows the protons to flow from the intermembrane space back into the matrix. So here we have the mitochondria and all these cristae here. So through the electron transport chain, the H+ concentration, assume all these dots are protons for H+, has increased in the intermembrane space. And now they will go through the ATP synthase in order to produce ATP. But then brown fat has this protein called uncoupling protein that allows easy leakage of protons back into the mitochondrial matrix, and by doing that they would increase heat production. And so brown fat is seen, for instance, in newborns and some hibernating animals, and is used as a mechanism to produce heat. Then, as we just promised, we're going to go over different forms of lipoproteins. We already discussed chylomicrons. What was the function of chylomicrons? They're secreted by intestinal epithelial cells and transport triglyceride to the peripheral tissues and cholesterol to the liver. So chylomicron, here you can see that it's transporting all the triglyceride to different tissues and then the cholesterol back to the liver. Now, what is the function of VLDL, IDL, or intermediate density lipoprotein, low density lipoprotein, and high density lipoprotein? So I've just made it a little easier here by showing you the pathways. So the function of very low density lipoprotein is to deliver hepatic triglyceride to the peripheral tissues. And so the triglyceride are being released by the lipoprotein lipase in the form of free fatty acids to the muscles and adipose tissues. And then any excess cholesterol is being transported back by the IDL to the liver. Now, what's the function of the LDL? So where is it? Oh, LDL right here. What's the function of LDL? So LDL, so you know how IDL transported cholesterol to the liver? LDL transports cholesterol to the peripheral tissues. And that's really bad, because as cholesterol goes and accumulates in the vessels, for instance, it could cause closure of these vessels and cause, for instance, heart attack or stroke or other problems. So that's why we don't want high LDL level. We don't want LDL to keep distributing cholesterol to the peripheral tissues. And then the final molecule we got here is HDL. What's the function of HDL? So basically HDL transport cholesterol from peripheral tissues to the liver. So all the cholesterol from peripheral tissues will go back to the liver, and that's great because we don't want cholesterol to build up in our vessels and other tissues. We'd rather send it to the liver where it will metabolize it there or convert it to bile acids for digestion of fat. So it's great. We always want to have high HDL level and low LDL level. That's our goal. All right. Now we can move on to some practice questions. What is the net ATP production under anaerobic conditions? Is it 1 ATP, 2 ATP, 3, or 4? And then the second question is, mitochondrial proteins are encoded by? Is it both nuclear and mitochondrial genes, mitochondrial genes, nuclear genes, or since they are maternally inherited, we do not require any gene transfer? So if you need more time, go ahead, pause this video, and once you feel ready, then you can resume the video. Alright, so the first question is pretty easy. Anaerobic condition, under glycolysis, we are just producing 2 ATP. Alright, now let me make the question a little more challenging. How many ATP, assuming that each NADH produces 3 ATP and each FADH2 produces 2 ATP, how much ATP is being produced by aerobic respiration? 36 to 38. So in which organisms do we get 38? So in prokaryotes, since they don't have any membrane-bound organisms, we get 38. And why is it that in eukaryotes we get 36? Because the 2 NADH that are produced in the cytoplasm during the glycolysis needs to be transferred into the mitochondrial matrix, and this process takes 2 ATP in order to transport 2 NADPH. So therefore, instead of 38, only 36 ATP will be produced. Now the last question that I want to ask you, out of this 36 or 38 ATPs, how many of them are being produced by substrate level phosphorylation? Substrate level phosphorylation. So we had 2 ATP that were produced during glycolysis, and then another 2 ATP that were produced in the citric acid cycle. So a total of 4 ATP are being produced by substrate level phosphorylation. The rest, 10 NADH2 and 2 10 NADH, and 2 FADH2, once they go through the electron transport chain, all the adenosine triphosphate that they produce are through the oxidative phosphorylation. All right. So that was for the first question. Now let's move on to the second question. If you want to think a little more, go ahead, pause the video. So mitochondrial proteins are encoded by both nuclear and mitochondrial gene. So let me give you some background information. So the primitive anaerobic cells, it's thought that from symbiotic relationship with an aerobic prokaryote, they have become a primitive eukaryotes, where the aerobic prokaryote is functioning as a mitochondria for energy production through aerobic respiration. So therefore, these prokaryotes have their own mitochondrial genes. They have their own genes. But then over time, over many years, they would eventually become dependent on the nucleus, some of the genes that are produced by the nucleus. And some of these genes actually do enter the mitochondria and function there. So therefore, mitochondrial proteins are encoded by both nuclear and mitochondrial genes. And so this one is not correct. Nuclear genes only is not correct. Mitochondria are inherited maternally, which is correct, but this part doesn't make sense, and they do not require any gene. Why? Why don't they require any gene? Because they are inherited maternally. We are already talking about being inherited, so where are their genes? So therefore, choice D is also incorrect. All right. And now let's complete this image. Regarding the primitive plant-like eukaryotes, there were also some photosynthetic prokaryotes that throughout the same process has started a symbiotic relationship with the eukaryotic cells, and they have turned into the chloroplasts. All right, now let's move on to the next set of questions. Which of the following statement is true regarding the uncoupling proteins? Which of the following hormones or molecules suppresses the glycolysis pathway? And then finally, acetyl coenzyme A participates in which of the following reactions? So if you need more time, go ahead, pause the video. Otherwise, we can proceed with answering these questions. So first one, which of the following is a true statement regarding the uncoupling protein? So they are found in high concentration in white? No, it's the brown adipose tissue. So therefore, choice A is incorrect. Preserves heat in newborns. No, it generates heat. It's not preserving. Preserving heat is the function of the white adipose tissue that provides an insulating sheet. But brown fat doesn't preserve heat, but rather generate heat. So the uncoupling proteins, they function by generating heat. Right? So just be careful of the wording. It increases ATP synthesis by promoting proton movement back into the mitochondrial matrix. So it's true, it promotes proton movement back into the mitochondrial matrix, but it's not through the ATP synthase. So therefore, ATP synthesis actually decreases because this protein equilibrates the mitochondrial protein gradient. And so therefore, the ATP synthesis would be reduced, and so choice D would be the correct answer. And here I have a drawing for you which shows you how all the H+ that is in the intermembrane space can be leaked back through the uncoupling protein back into the mitochondrial matrix, as a consequence of which there would be generation of heat. Alright, next question. Which of the following hormones or molecules suppresses the glycolysis pathway? So again, I didn't want you to memorize all these allosteric effectors. I still don't want you to memorize it. I think you can work it through. Alright, so glycolysis. What is the end product of glycolysis? It would generate NADH, it would generate ATP, and then later on acetyl coenzyme A will be produced from pyruvate acid, and then it would increase the citrate production through the tricyclic acid. So any of these molecules, any of the products of the glycolysis would suppress its pathway. So right away you can see that choice B is the correct answer. Now let's see why the other choices are incorrect. Well, if AMP or ADP, if the concentration of adenosine monophosphate or adenosine diphosphate is increased, it means we don't have enough energy. So we want to enhance glycolysis in order to increase the ATP production. So there we go. These two are incorrect. How about insulin? Well, the function of insulin is to decrease the blood glucose concentration by any means possible, increase the glycogen synthesis, increase the lipid synthesis, or increase the glucose breakdown, which is glycolysis. So insulin will enhance glycolysis, it won't suppress it. So therefore, that is also the incorrect answer. And so choice B is the correct answer here. All right, next question. Acetyl coenzyme A participates in the which of the following reactions? So do you recall glucose through the glycolysis gave rise to pyruvate, and then this gave rise to acetyl coenzyme A, and now acetyl coenzyme A can enter the citric acid cycle. So participates definitely in the citric acid cycle. Acetyl coenzyme A is one of the end products of the glycolysis, but it doesn't participate in the glycolysis pathway itself. So that's why this is incorrect. Fermentation is where pyruvate is being converted into either ethanol or lactate. And so again, acetyl coenzyme A is not directly participating in fermentation. It's the pyruvate that is participating. And then finally, electron transport chain, acetyl coenzyme A contributes to the production of NADH and FADH2, which could be used by the electron transport chain, but it doesn't directly participate. Have we said cytochrome C or ubiquinone, then I would say yes, those do participate in the reactions, but acetyl coenzyme A does not directly participate. And so here is a refresher from the first video that we had. Pyruvate via the aerobic respiration, it gives rise to acetyl coenzyme A that can go through the citric acid cycle. And so that's why Kreb cycle or citric acid cycle is the correct answer here. Alright, now the final sets of questions. Acetyl coenzyme A acts as a building block for which of the following molecules? And then, which of the following is a true statement? Go ahead, pause the video if you need more time. And then we have one more question. Which of the following is the correct order for recipients of electron in the electron transport chain? So once again, if you need more time, go ahead, pause the video. I'm going to start providing the answer for you here. So acetyl coenzyme A, we already discussed it's required for ketone body formation, and then we also discussed that it's required for fatty acid synthesis as well. The only thing we didn't discuss is the amino acid synthesis, and so therefore choice B is correct. And if you recall, amino acid is made of a central carbon, so this is a unit of the amino acids, an R side group, amino group, and a carboxyl group. We are going to, in a separate video, discuss the structure of proteins and amino acids, but acetyl coenzyme A, which is a 2-carbon molecule, is not one of the building block for amino acids. All right. Next question, which of the following is a true statement? The majority of glycolysis reactions take place in the mitochondrial matrix. No, we already discussed it takes place in the cytoplasm. That's why the 2 NADH that are produced from glycolysis, it takes some energy to transfer them to mitochondrial matrix. So choice A is incorrect. Solid tumor cells have a low rate of glycolysis. So what happens with the tumor cells is that they lack an extensive capillary required for feeding oxygen to the tumor, as a consequence of which tumor cells are mostly surviving under hypoxic condition, and so therefore they have high rate of glycolysis, not low rate, high rate. And then for the same reason, they don't need a large number of mitochondria. No, they just need a short number because they're going through glycolysis. Why do they need mitochondria when they don't have oxygen? So choice C and choice D are also incorrect. The rate of glucose consumption is higher under anaerobic than aerobic conditions. And this is correct because how much ATP is produced from anaerobic respiration? 2 ATP. And how many from aerobic respiration? 36 to 38, assuming 3 ATP per NADH, 2 ATP per FADH2. So therefore, one glucose molecule will yield many more ATPs under aerobic condition. But under anaerobic condition, there's so little ATP production that the glucose consumption has to keep increasing to compensate for that. Alright, and then the last question. Which of the following is the correct order for the recipients of the electron in the electron transport chain? Right, if you forgot, I here brought you the image again. So it's the NADH and FADH2 that pass on electrons to the ubiquinone. So here, NADH and FADH2 pass on electron to ubiquinone, which will pass on electrons to cytochrome C, and then finally cytochrome C passes electrons to the oxygen, which is the final electron acceptor. And so therefore choice B is the correct answer for this question. Alright, and so that's about it. I hope that you found these 2 lectures on metabolism of the glucose and energy production useful. and thank you all for listening.